Hello, everyone. My name is Manoj, and I'm a PhD candidate at the University of Sydney and Children's Medical Research Institute. So today I'm going to be talking about one of the works which I've been doing in the past few months. It's almost complete, where we have developed an interpretable deep generative framework for capturing cell identity, along with an automated annotation tool for single-cell multi-omics data. So before I go into framework, I just want to give you a little bit of background. As you might know, traditionally bulk sequencing has been used to understand tissue level heterogeneity. And however, the problem is it only captures the global or average expression profile across tissue and fails to reveal cell-to-cell variability. That's where single-cell omics comes into picture. Single-cell omics helps us understand the molecular variations occurring in each individual cell, thereby providing a high-resolution view of cellular heterogeneity. Even though single-cell omics is advantageous in this, analyzing single-cell omics data is not an easy task. So often single-cell omics data is high-dimensional, which means for each individual cell, you have thousands of genes. And the data is quite noisy, the dropout events, and it's often sparse, which means majority of the expression values will be zero. So a number of tools have been developed, including statistical-based methods, classical machine learning-based methods, and recently deep learning-based methods. Now, deep learning-based methods are particularly well-suited for analyzing these sparse and high-dimensional single-cell data, and they are quite good in capturing these nonlinear relationships that that often appears in this single-cell data. However, one of the problems with deep learning models is their lack of interpretability. Even though they are so good at analyzing this data and making predictions, they often understanding the reasoning behind their predictions is hard. So a number of tools have been developed, particularly in the last 2 to 3 years, to address this issue. And we have done a comprehensive review on this, particularly for single-cell omics data, and it's now out in bioinformatics. So please feel free to check that out. So building upon this, we developed an interpretable framework, which we call Hydra. So the idea behind this is that we are using a variation autoencoder. We are conducting a specialized training. So here I have basically simplified the framework as the work is currently unpublished. And we do multitask learning. So your input can be your single-cell RNA-seq data, single-cell ADT, like surface protein, or single-cell ATAC, chromatin accessibility data. So you can provide them as individual modalities, or you can provide them as joint modalities like site-seq, share-seq, TA-seq, and many other technologies. So basically, our version encoder will be trained on this original single-cell data. And once we do this, we are going to generate balanced data sets using our model by randomly downsampling the major cell types and producing new minor cell types. Then finally, we fine-tune our models, and we do this multiple times to get an ensemble of models to capture the cell identity genes. Once we do this, we are going to use this further in our automated annotation framework, which consists of a simple ensemble of simple neural network classifiers, which helps us in automatically annotating the cell types. We validated our method on, I believe I validated on 22 or 23 data sets and 11 different methods. So I'm going to talk about the results now. So one of the first things that I want to talk about is the stability of the features. So what I mean by stability of the features is that when we are learning the cell identity genes, we want to select consistent set of features. What I mean is a small variability in the data set should not result in your model capturing completely different set of genes. So that is not really useful in real world scenario, because we often don't train every single time. We train on a reference data set and we use that to automatically annotate. So this is why feature stability is quite important. And we do that by computing the Pearson's correlation coefficient. So here I'm comparing against 4 well-known statistical methods. And the reason specifically I'm comparing against statistical methods is that previously we did a benchmarking on evaluating this, and we found that deep learning models often have a little bit lower reproducibility compared to statistical models. So this paper is in Genome Biology. It was published last year, so feel free to check that out. So one of our ideas was, can we use ensemble to improve this? And we found that, as you can see, now our model, especially in interdisciplinary feature stability, our model is performing quite well compared to other statistical models. So here I'm showing you the 3 smallest and 3 largest cell types for the prostate urethra dataset. So next thing is capturing the cell identity genes. So here what I've done is, from the mouse cell atlas, I have taken one particular cell type. In this case, I have taken the kidney PCT epithelial cell. So basically what I mean by cell identity genes or cell type specific genes is that when we select these genes, they should show expression specific to our cell type of interest. So here I have chosen top 5 genes provided by our model. And basically the red color in the first one, what you're seeing is the log normalized expression. So if it's cell type specific, we want our genes to show expression specifically in our cell type of interest, that is the kidney PCT epithelial cell. And as you can see, which is what we are observing. And on the right side, next, what I did is whether the expression is highly specific in this cell type compared to all other cell types. So here I have done a ridgeline plot, and as you can see, it's exhibiting the same thing, that the genes provided by our model is specific to this cell type. So this is very important in capturing these cell identity genes. So here I have done for so the previous one, I selected top 5 genes in one particular cell type. So here now what I did is I created a similar data set with 20 cell types randomly chosen, half as major and half as minor, with an imbalance ratio of 100:2. So basically, we would be, each of these rows would be like cell types. So basically, I'll be taking genes belonging to this particular cell type and checking the expression in this. So basically, it should show dark blue color in this and not in anything else, unless the cell types are related. So when we keep doing this, we ideally want to get this very clear mosaic pattern, and that's what exactly we are observing here. So our deep learning model is able to capture the cell identity genes like the statistical models. Next, I want to talk about annotating cell types from single-cell transcriptomic data. So what I'm showing you here is intra-dataset cell type annotation, where you would split your dataset into train and test. So here I'm doing 5-fold and saying, so here I'm showing you the UMAP of the ground truth and hydra. As you can see, we are able to annotate most of the cells effectively. But this might not tell you how good it is in comparison to other methods. So this is the plot for that. Where all the cell types are arranged from smallest to largest. And as you can see, I'm comparing against 4 other methods. And these methods, they often perform quite well for the major cell types. When it comes to minor cell types, the smaller cell types, they often suffer due to dominance by major cell types. And here I have included the overall performance, and we seem to perform quite well compared to all of these very well-known methods. Next, what I have is interdataset cell type annotation. So what I mean is, in real life, in real world scenario, sorry, we wouldn't actually train every single time. So train on reference and test. So here, interdataset could be evaluating on different batches, different datasets, different generated by different labs. Right, so here I have evaluated on 25 trained test pairs across 4 tissues: kidney, lung, PBMC, and retina. And all the methods have been ranked based on the overall accuracy. As you can see, we are performing quite well compared to other methods in terms of annotating cell types. Now moving on from transcriptomic to multimodal. So first, what I'm showing you is, again, interdataset annotation. So SciCAR, which is jointly profiling RNA and ATAC. So here I compare against 3 well-known methods: MOFA+, Human, and SMOMAN. Again, the same case, as you can see, we are keeping the consistent performance, while these perform quite well for the major, but often poor. And this is the overall cell type annotation performance. And this is another one, SNARE-seq, which is again RNA and ATAC. Finally, we have the interdataset cell type annotation, where compared across 26 trained test pairs and 3 technologies from 4 different studies: 10x multi-ohm, SiteSeek, and TSA. And as you can see, we are at 74%, while the second best performing method is at 60, and other methods are quite performing poor. That's mainly because they have poor performance on the minor cell types. So yeah, in summary, we have developed a single unified framework for capturing the cell identity genes, along with an automated annotation module. One of the advantages is that it provides interpretability due to cell type specific features, and allows integration capability with diverse single cell molecule modalities. So finally, I would like to thank my supervisors, Peng Yang and Alice Patrick, as well as my colleagues at Children's Medical Research Institute and Sydney Precision Data Science Center for their valuable feedback, which has really improved my work. Thank you very much.